In the last lecture, we left off talking about the termination of replication, right? So I think that the number here was termination 3. Okay, so what I have here is, here's a replication bubble, and let's imagine that replication is occurring. We'll have the newly synthesized strand in red. So if this was the original origin, we start with the original primer, then this would be 5', and this would extend towards the fork. So that would be the continuous synthesis. Now at the opposite fork, this would be going in the away from the fork. So that's discontinuous synthesis. On the bottom strand, if we put our primer down here, this would be the 5' end of the primer. And so on the bottom strand, on the left-hand side, that's the continuous synthesis. This one would be going away from the fork and be done in segments called Okazaki fragments. So in the termination of replication, as I said, termination occurs in a circular molecule or in internal portions of a linear molecule when new strands run into each other. The new strands run into each other, they meet, polymerase leaves, and then DNA ligase seals the nick. But at the end of linear strands, so right now we're talking about the end of linear strands. What happens? So let's just imagine this was this very short segment, and we zoom ahead in time until we finally get to the end of the strand. So here is polymerase chugging along in continuous synthesis, until it falls off the end here. And so synthesis is complete. We've got a fully synthesized strand. What about this lagging strand, however? This, remember, was done in chunks. So if this was one Okazaki fragment, oops, sorry, here's another Okazaki fragment. Polymerase with DNA polymerase 1 would come in here. Sorry, let me kind of clear this up a little bit. DNA polymerase 1 would put down, would come in, run into the other primer, replace it, right? And so ligase would then seal the nick. But look what happens here. This primer that's put down, primase doesn't know where the end of the molecule is, right? So this is our RNA primer. Primase does not know. So primase doesn't line up perfectly with the end of the molecule. And in addition to that, remember, this is an RNA segment. And it can base pair with DNA, but it's not as stable. So what's going to happen is little nucleases are going to come along and they're going to chop up the linear ends. So in linear chromosomes, the lagging strand shortens with each replication. And so remember, replication occurs every time a cell divides. So lagging strand shortens with each replication. So every time DNA replicates, the lagging strand is going to shorten. Now over here, this is going to be the lagging strand, so this end would shorten. Now, does that sound sustainable? You know, if, say you've got a gene right here, eventually after enough replications, you'd start cutting into the genetic material. So this is not a sustainable situation if we didn't have some mechanism to protect the ends of the chromosomes. So there is protection at the end of the chromosomes, and those are called telomeres or telomeres. These are multiple copies. Now that could be, you know, 100, 150, even up to 200 copies of short. In short, again here, you know, 5 to 7 base pairs of short DNA repeats. In humans, in fact, the repetitive sequence is 5' T, T, A, G, G, G. So at the end of human chromosomes, at the end of linear human chromosomes are these, 100 to 150 copies of TTA-GGG, TTA-GGG, TTA-GGG. And what that does is it protects the genetic information. Let's take a look at an image here. TTA-GGG, TTA-GGG, right? So how did these get here? How are these telomeres created? And even after a certain amount of time, if they were only created once and they continued to shorten with every cell division, ultimately they would shorten out of existence, right? So there's an enzyme that creates the telomeres. I'm gonna erase this slide now. So the enzyme that creates telomeres or telomeres is called, cleverly enough, telomerase. Now think about this. This is an enzyme. This is an enzyme that synthesizes the telomeres. So it's an enzyme that synthesizes telomeres, and it's really kind of interesting because it's not just a proteinaceous enzyme. So it's not only a protein, it's a polymerase enzyme activity plus associated with this telomerase, excuse me, is an RNA template. So a single-stranded RNA template strand. And this is going to serve as the template to create that TTA-GGG. So let's look at the image again. So here, this green looking blob is the protein polypeptide component with the polymerase activity. But look at, as part of the telomerase enzyme is this single-stranded RNA template, which is complementary to this repetitive sequence, to this repetitive TTA-GGG. So telomerase comes in, it will lengthen and create this telomere to, like I said, 100, 150 copies of this sequence, and comes in and it lengthens it. And that way it creates this sort of buffer zone at the end of linear chromosomes. Let's take a look at telomerase animation.
Meet Suvi, the magical all-in-one countertop kitchen robot.
Just a second here. There we go.
The ends of chromosomes contain regions referred to as telomeres. These telomeric regions consist of telomeric repeat sequences. The exact sequence of the telomeric repeat can vary from species to species. The repeat sequence shown here is found in the telomeres of human chromosomes. Since DNA replication can only proceed in the 5' to 3' direction and requires a primer, it is not possible for DNA polymerase to replicate the 3' ends of linear chromosomes. If this problem were not corrected, chromosomes would shorten with each cycle of DNA replication. The addition of telomeric repeat sequences by an enzyme complex referred to as telomerase assures that this shortening does not occur. Telomerase is a unique and interesting enzyme because, in addition to having DNA polymerase activity, it also contains an RNA sequence that provides a template for the synthesis of telomeric repeat DNA. Part of the RNA sequence hybridizes with the single-stranded overhang of the DNA strand, leaving a single-stranded overhanging RNA sequence. The repeat unit shown here is from tetrahymena, which is a model organism used in telomere and telomerase research. The DNA polymerase function of telomerase then synthesizes the DNA strand complementary to the RNA found in telomerase, and subsequently translocates to the end of the newly synthesized strand, and the process repeats itself. Many cycles of repeats can occur. Once telomerase has completed its function, DNA primase synthesizes an RNA primer near the 3' end, and DNA polymerase fills in the vacant region. A short region at the 3' end will remain single-stranded. The end result is that telomerase will have added many repeat sequences, from a few dozen to several hundred. This prevents chromosome shortening.
So that's how the telomeres are created. So we can think about this. I'm just going to put in here. At the end of linear chromosomes, there's a sort of buffer zone, which are dozens to hundreds of copies of that repetitive sequence. Now, telomerase and the telomeres are actually important for a couple of important health issues in life because, so telomerase, let's think about this, the protein component of telomerase, where does telomerase come from? Do you have a telomerase sandwich for lunch? Well, you might eat some telomerase and if you have a hamburger or something, but that's not what's working in your cells. Telomerase is coded for by a gene within the human genome. And there's also some associated proteins that go along with it. But genes are not always expressed at all the time. And so telomerase is not always present in cells. So telomerase is expressed or present or on or active in early development. So in a developing embryo, fetus, so in development when there's lots of cell division going on, and in stem cells. So stem cells are undifferentiated or unspecialized cells that have unlimited potential to divide. So unlimited division potential. So that makes sense. So cells that divide a lot, hundreds, thousands of times, they need to have those telomeres continuously refreshed, right? So telomerase is on in those cells. But fully differentiated adult cells, so differentiation is the process of a cell taking on more and more specialization. So differentiated adult cells do not express telomerase. So you can think about telomerase as being off. So when cells are fully differentiated, so now we're talking about a skin cell, a white blood cell, a liver cell, a heart cell, a neuronal cell. When a cell is fully differentiated to its final form, it quits expressing telomerase. So that means each time the cell divides, the telomeres shorten. So telomeres shorten with each cell division. In effect, the length of the telomeres can act as a cellular biological clock. The shorter the telomere, the more times the cell has divided. It's a way to kind of keep track of how many times a cell is divided. So they shorten with each division and acts as a cellular biological clock. When they get too short, when they shorten too much, when they're almost gone, when they're too short, the cell undergoes apoptosis, which is also known as programmed cell death. The cell kills itself. It says, you have lived your life. It is time to go away, right? Let the new cells come in and fill in the area. So this is a natural sort of biological clock. So telomeres are important for aging, right? So understanding how cells age and the aging process, telomeres are a key component of that. And it's also a key component to one of the, the most important health diseases in the world, because it is also upregulated in cells that have uncontrolled cell growth. So can you think of a disease, or maybe 150, 200 different types of diseases that result in uncontrolled cell growth? Cancer. So in cancer, cancer isn't just one disease. There's hundreds of different types of cancer. But what you can find in most types of cancer, if you take those cells and you look at telomerase activity, telomerase is often re-expressed in tumor cells. because of in cancer, right? Unlimited division potential. The cells are growing uncontrollably. So the telomeres have to be refreshed or the cells would kill themselves when they shorten too much. So telomerase is also a huge area of research in cancer cell biology. It's not the only thing mutated or present or being abnormal, but it's one of the key factors that is often found in cancer cells. So that's what happens at the end of linear chromosomes. We've got these telomeres protecting our genetic information. We've got telomerase, the enzyme that makes the telomeres. Let's see, what else do we want to talk about with replication? We'll just briefly mention the rate of replication. So rates of replication vary depending on the organism. And so we'll look at the rates in prokaryotes and eukaryotes. And we actually looked at this in a problem set. So prokaryotes have a very approximate replication rate of about 1,000 base pairs per second. Now think about eukaryotes in contrast to prokaryotes. What do eukaryotes have far more extensively than prokaryotes? We have a lot more DNA, but we have multiple origins of replication to help kind of tackle all that genetic content. But that DNA is wrapped around histones, is packaged into chromatin. So replicating eukaryotic DNA, you've got to get those nucleosomes out of the way so that you can free up the bases to act as a template. So eukaryotic replication is much slower, and it can go in the range from 10 to say 100 base pairs per second. And because it's packaged in chromatin, that chromatin has to be sort of remodeled or pushed out of the way. The proteins have to be pushed out of the way to allow for the replication process to occur. So that slows things down. Now, remember, the human genome is like 3 billion base pairs. And so think about the feat of adding 3 billion base pairs by polymerase, adding in those complements. They're only matched up by hydrogen bonds, which are weak interactions. So it's conceivable that we could have errors during replication. And so let's imagine, let's make a template strand of DNA. So we'll say this is a 3' end, and then we've got our newly synthesized strand coming in, CTAA, going in this direction. So now it's conceivable that the wrong base could get into this spot. And if that occurs, let's say it's a C. And then what we have here is a mismatch. It's called a mismatch, error of incorporation. And it occurs with a frequency, with a reasonable frequency. So approximately every 1 in 10,000 base pairs. Now, if that were the case, if you think about that, our DNA would be riddled with mistakes if we had one mismatch per 10,000 base pairs. So actually, DNA polymerase has a mechanism to correct that, which reduces this error rate dramatically. So there's a lower actual or realized errors, and that's due to a specialized function of polymerase. And we call it the proofreading function. Activity of DNA polymerase 3. DNA polymerase 1 also has this function, but DNA polymerase 1 really doesn't synthesize that much DNA. So in the bulk of proofreading that occurs is by DNA polymerase 3. So what happens with proofreading? So imagine here's our DNA polymerase enzyme. It's chugging along 5' to 3'. What happens in proofreading function is that polymerase recognizes the error. This is going to have a confirmation on no matter what the mismatch is, it's going to be topographically, morphologically, it's going to not be normal, right? And so it is recognized, so the error is recognized. Oops. By the polymerase, by DNA polymerase. Synthesis stalls. So there's a pause. And it backs up. So here's on our new strand, we've got 5' to 3'. So it's recognized, the synthesis stalls, and DNA polymerase is going to back up in a 3' to 5' direction and cut out the error. So the error, so the polymerase backs up in a 3' to 5' direction and removes, cuts it out. What kind of enzyme cuts phosphodiester bonds? Using a 3' to 5' exonuclease activity to remove the error and then it replaces it with the correct base with its 5' to 3' polymerase activity. So here is an image of that. Here's our mismatch of incorporation. Here's the growing strand in a 5' to 3' direction. This gray box is supposed to be depicting polymerase. Well, the error triggers or causes the polymerase to stall. It then backs up. It removes this error using the 3' to 5' exonuclease activity. And then it puts this back in with phosphodiester bond using its 5' to 3' polymerase activity. So if you remember primer removal by DNA polymerase 1 used a 5' to 3' exonuclease activity. Proofreading uses a 3' to 5' exonuclease activity.